Hello class, welcome back to our new class. Today in today's class we are going to see the humoral immunity. In the last class we have seen the cell immunity. In today's class we are going to see the humoral immunity. Now, the humoral immunity is defined as the immunity mediated by antibodies which are secreted by the B lymphocytes. We have seen that the cell immunity or the T cells immunity. Okay, so in this immunity, there are antibodies which are secreted by the B lymphocytes. The B lymphocytes they are secrete the antibodies into the blood and the lymph. The blood and the lymph are the body fluids, and since the B lymphocytes provide immunity through the humors, this type of immunity is called as a humoral immunity. तुमचा जो ह्युमर नावाचा बोन आहे त्यामधनं हे lymphocyte तयार होत असतात. So that's why it called as a humoral immunity. The lymphocytes provide immunity through humors. So that's why this immunity is called as a humoral immunity or the B cell immunity. The antibodies are the gamma globulins produced by the B lymphocytes. And these antibodies, they are fighting against the invading organism or the foreign particles which are entering into the body. The humoral immunity is the major defense mechanism by bacterial infections. In the case of cell-mediated immunity, the macrophages and the other antigen presenting cells play an important role in the development of humoral immunity. So I already told you in the first lecture that they are working in collaboration. The T cells and the B cells they are working in the collaboration. So both the immunity types of immunity they play a major role in the development of immunities. Now, the role of antigens they are presenting in cells. The injection of foreign organisms and digestion of their antigens by the antigen presenting cells they are explained by the presentation of antigens. The antigen presenting cells they are present into the antigen products bound with the HLA proteins which are present in MHC2 molecules. MHC2 protein molecules to the B lymphocytes. Then this activates the B cells through the series of events. We will see the sequence or the series of events they are occurs during the activation of B cells. The B cell they recognize the antigen displayed on the surface of the antigen presenting cells. Then with the help of its own surface receptors they call as a B cell receptors. The recognition of antigen by B cell initiates a complex interaction between the beta cell receptors and the antigen cells. And this reaction they are activated by these reactions they activate the B cells. At the same time, the macrophages or the antigen presenting cells release interleukin 1 which facilitates the activation and the proliferation of beta cells. Then activated beta cells, activated B cells they are proliferate and the proliferated cells carry out the further actions. Simultaneously, the antigen bound to class 2 proteins molecules activate the helper T cells and also resulting in the development of cell-mediated immunity. Then these B cells they were transformed into two types. एक होता plasma cells and the second will be the memory cells. The role of plasma cells in case of B cells, the plasma cell destroy the foreign organisms by producing the antibodies. Antibodies are globular in nature. The rate of the antibody production is very high. That is, each plasma cell produces about 200 molecules of antibodies per second. The antibodies they are also called immunoglobulin proteins. The antibodies they are releasing into lymph and then transported into the circulation. Then the antibodies they are produced until the end of lifespan of each plasma cells, which may be from several days to the several weeks. Then a role of memory cells. The memory B cells occupy the lymphoid tissue throughout the body. The memory cells, they are in active condition until the body is exposed to the same organism for the second time. During the second exposure, the memory cells, they are stimulated by the antigen and produce more quantity of antibodies at a faster rate than the first exposure. The antibodies produced during the second exposure to the foreign antigens are also more potent than those produced during first exposures. And this phenomenon forms the basic principle against the infections. Then we have seen the role of Th cellular T helper cells. The T helper cells are simultaneously acted by antigens. The activated T helper cells secrete two substances called interleukin 2 and the B cell growth factors, which promotes the activation of number of B lymphocytes. They also helps in the proliferation of plasma cells, and they also helps into the production of antibodies. So this is a humoral immunity which activate only when there is a activation of B cells, and there are the certain sequences which are come into the view for the activation of B cells. Both the immunities, that will be the cell immunity and the humoral immunity, they will work in collaborations. One immunity cannot perform 100% in the absence of other kind of immunity. So these B cells and the T cells, they work together in such a way that these the foreign particles which are coming inside the body, they will destroy by this T and the B cells, which plays the major role in case of the immunity of the humans. In next class, we are going to see the ELISA techniques which are used for the immunity. Until then, I'll see you in next class. Again, from 14, you will be having your exam. So this will be the last class of your semester 6. I hope you have gone through the all lectures. The leather technique PPT I will be uploading onto your on your what we can say on your Moodle site. Please go to the all lectures. It will help you in your exams. Okay. Okay. I will see you in next class. Until then, see you.